Okay, thank you. Am I too loud or you can hear me fine in the back? Thank you. Thank you, the organizers, Dr. Bob, Jessica, for your kind patience in inviting me to speak. And
Thank you for speaking.
thanks to the grad students, organizers, and thanks for the introduction. So I'm looking at the title, I'm like, what am I, why did I even contribute this title? And because it's not a one path or a clear path or any kind of a path that I'm going to tell you today. And what I'm going to share with you is not a typical research seminar. It's a combination, there's a lot of things in here. So what I'll say to you is that do not be stressed out. Just relax, take a break from the hardcore plant breeding seminars that require a lot of thinking and critical analysis of data to something easy to digest, and hopefully the last slide should be what you should take home with you. So I hope to accomplish that. The man who fed the world, I'm not standing here even trying to compare, even trying to dream. I can just say that there's also men and women and you plant breeders that are involved in feeding the world. And I would like to show a video here, and if this works out, you should hear us.
All my friends think I just grow plants for a living, but that's only part of the story. As plant breeders, we increase the resistance of our crops to a whole slew of biotic and abiotic factors. Drought, frost, pests. I'm mostly interested in this little guy right here, the culprit for northern leaf blight and maize. Hard to believe this fungus can cause yield losses as high as 70%. It's a huge problem for maize-dependent areas like Africa, where food security is already an issue. My goal is to dissect the genetic mechanism behind disease resistance and incorporate it into maize varieties. It's definitely a challenge. Want to hear stats like this?
Our latest estimates show that there are 925 million black people in the world in 2010.
And see population projections like this. It's hard not to be motivated. Dude, 9.1 billion? That's a lot of mouths to feed. But challenge accepted. Everyone is entitled to food. And breeding crops that are more resilient and adaptable to changing climate is more important than ever. My name is Ariel Chan, plant breeder and geneticist in training, working to feed the world.
Okay, so that's a grad student from Cornell, and I know they're very great and very competent graduate students at Iowa State, and I believe that we can compete at the same level. So Borlaug had to know a lot of things, not just breeding, a lot of other things. And I believe that today we need to collaborate, and as I say, the future is men and women. It's going to be men and women who fed the world. So again, I say, I warned you about my talk. I'm going to share about myself. I like to talk about myself. I'm from Tanzania originally, a small country in East Africa. That's where the red arrow here shows here the northern part of Tanzania. Behind these mountains is where I was born. Of course, moved away from this part of the world and obtained my undergraduate education at Africa University in Zimbabwe in agriculture, natural resources, and I like to learn about animals, so I specialize in animal science. After that, I worked for the United Nations, first in Angola for 2 years. Angola was at civil war at that time. It was interesting to work there. And when things got a little bit dangerous, when some of my friends were killed in some of the ambushes, I decided to leave Angola and go back to Zimbabwe, where I joined UNICEF. My work with the UN was food security, vulnerability assessment and mapping. So we would go to the villages and conduct food security assessments, look at what kind of resources they have, food, crops, animals, and say, okay, I think they're okay for this year. We come back to the program unit and say, these are the people that you should target and give aid to, food aid. So I made decisions of who gets food and who doesn't get food. So during this time, I came across a lot of, even though I'm from Africa originally, I had not appreciated this suffering with the hungry people, desperate people, until I started now, after undergraduate education, starting to process it. Coming across these female-headed households where the woman is raising 10 kids. I mean, the husband is left for mining work in South Africa. or has died of AIDS, and they have 10 kids to raise. And so one of the days I was visiting a village in Zimbabwe and with UNICEF, and I came across this household, and this lady came and invited us to come in and looked at the number of chickens she had and goats and other things. And in the middle of the ground there was a 2-year-old who had a plate of porridge. This little girl was eating the porridge that was from UNICEF. And I came near this girl and she looked at me and she smiled. And that's the day, to me personally, I think I made a decision that I would stay doing this kind of work for the rest of my life. I continue, I remember that child. I don't know her name, but I still see her in the back of my mind. I came to the US. I learned that I need to go back to grad school, get more education to be able to make an impact, and came to the US. I attended Murray State University, where I got a degree in agriculture at master's level, soil fertility. And then during this master's degree, I learned about biotechnology, agricultural biotechnology. I said, well, this is cool, recombinant DNA technology. This will be good for food security. We can change crop, traits of importance. So I was excited, and so I went to graduate, to PhD education at the University of Kentucky to learn this new technology. A little bit naive there, but I was excited. Anyway, I ended up getting my PhD from Nebraska-Lincoln. In plant breeding and genetics, specializing in plant molecular physiology. My work at UNL, I worked under Dr. Paul Staswick, a great plant biologist who studies hormone signaling. And my work, as the title here of my dissertation, is jasmonic amidosynthetases and their role in the plant wound response. So jasmonates are defense hormones that are involved in many functions in plants. And Staswick Lab had discovered this small gene family in Arabidopsis. Some members conjugate IAA, metabolize auxin, and some work on jasmonic acid. And my work was to focus on this gene that he had discovered and characterizing a tomato gene that was not, whose function was not known at that time. And JA amidosynthesis carry out this reaction here. Simply, they add an amino acid to a carboxyl group of a free hormone, such as jasmonic acid, to form a conjugate. And so one of the conjugates that now actually is the kind of paradigm shift in the field of hormone signaling in plants is that the free hormone was thought to be the active signal. But what Staswick discovered was that it was the amino acid conjugate that was the active signal, and that was jasmonic acid isoleucine. And it's produced through the Mevalonate Pathway, starting with alpha-linolenic acid in the chloroplast, through a series of reactions, including beta oxidation, to form the first isomer, the plus 7 isojasmonic acid, which is unstable and quickly forms the more stable form, the minus form. And of course, jasmonic acid can also be modified by addition of a methyl group to become a volatile molecule, the sweet scent of the jasmine flower that you have all probably experienced. But my work, as I said, was about the conjugate J-isoleucine and the JAR1 gene that encodes an enzyme that catalyzes this reaction here. So what is the wound response? I was fascinated by this, and I'm still fascinated by this. And of course, personally, I'm scared to death by these organisms here. If you bring one of these, I'll just run out of this room. But plants have evolved defense mechanism dealing with and interacting with organisms such as this. And so the organisms are evolving, and the plant is also evolving. It's an ongoing battle. But here's a summary, and it's information from the solanaceous, the groups that have worked on tomato, for example, the Bud Ryan group at Washington State University. You have this herbivorous organism feeding on the plant, so damaging it and adding its saliva compounds to the wound site. And that triggers the production of jasmonates quickly, followed by induction of defense genes locally and also systemically. And in some cases, it's been documented that the volatiles that are released due to this induction of the defense response attract natural enemies that prey upon this organism here. So this tritrophic interaction that is really interesting. And of course, the parasitoids lay their eggs in this victim here, and after some time, their kids are coming out of these larvae. And so yeah, that's quite interesting. Plants calling for help. And of course, plants that fail to produce jasmonic acid get decimated by such herbivorous organism. You can see here ACX1 is completely defoliated compared to wild type. And the larvae growing on the wild type plant do not perform very well through their growth, but those in ACX1 grow vigorously because the defense response here involves production of proteinase inhibitors that interfere with digestion of nutritious stuff that they get from the plant. So they get sick. So I didn't do this work, and I don't want to do it. This is just recently that mosquitoes also perceive jasmonate and they don't like it. Yes, I didn't touch insects. I didn't work with mosquitoes. My work was simple. It was a simple assay involving wounding a leaf, mechanically crushing it across the midvein, and monitoring the expression of the JAR1 gene, followed by detection of the conjugate J-isoleucine. And I had wild type and a mutant to compare. And I want you to note that the mutant plant that we knew it produced an enzyme that was not functioning was still producing low levels of the conjugate. That became a heartbreaking situation for me, but I think it was a blessing in disguise because I could get seed out of those plants. I didn't see it then. Anyway, looking at the induction of defense genes, which was a disappointing place for me, was that we didn't see a dramatic impact when we looked at wild type compared to JAR1. So genes that depend on the jasmonate signal for their induction in response to wounding. There's a slight delay here in the induction of VSP2. We successfully isolated the tomato JAS, some molecular biology here, making recombinant protein and expressing in E. coli cells, conducting some biochemical assays, looking at which of these isomers the enzyme was active on. And I just, here a summary, this work is published, I don't need to tell you too much about it, that there's high specificity for isoleucine and a high preference for this isomer here and not the other. We attempted to silence the gene, and again, the knockdown approaches don't give you a complete elimination of the transcript. But we have two transgenic events, independent transgenic events here, where we are silencing the JAR1 from tomato by RNAi approach. Apply wounding, the gene is induced in wild type and low levels in the RNAi lines, and the conjugate also is suppressed in these lines. So that's a summary of what I did for jasmonate part. And actually, this is the part that is more interesting. Our work, during this time and out of Staswick lab, John Browse and Greg Howe at Michigan State made a huge discovery that went to Nature, that J-isoleucine is truly the bioactive signal. I missed it, so yeah, things happen. But we contributed significantly to their work, and that was the key for you graduate students. Remember that. Even if you're not the superstar, you are part of the stars. And so I think that was cool, that now we have an idea of how the signaling occurs inside the cell, jasmonate signaling. And then during my graduate education in Nebraska, I developed interest in sterols. And this was out of being asked by my committee to write an NSF proposal as part of my preliminary. So I wrote about sterols, and I learned quite a bit about them, and that they are produced through the Mevalonate Pathway. Of course, you know this, and we knew from our cell biology classes. We know from plants that they are precursors to brasinosteroids, and their specific functions in plants that are of interest. So I went for my postdoc at Kentucky and worked with Joe Chappelle, who is an expert in isoprenoid metabolism. And he asked me to use Nicotiana benthamiana as a model to ask this question: how is this pathway regulated in plants? We knew how this pathway is regulated in animals, in us. So one of your liver cells right now is facing a challenge of balancing de novo production of cholesterol versus cholesterol that is coming from what you've ate in the afternoon, from your diet, through the receptor-mediated intake into the cell. And that happens through a feedback mechanism, so cholesterol feedbacks and shuts down important genes, especially HMGR, which is a key control point for this pathway. So we know about this in animals, and the question is about plants. The mevalonate pathway, the upstream part is conserved, but when you come to the formation of these cyclic molecules. You see this bifurcation here where you have only one sterol produced in mammals, one in fungi, but several in plants. Which of these feeds back, if there's a feedback regulation of any sort? So and again, plants also do this kind of decorating the molecules by adding methyl ethyl group at carbon number 24, and which is only happening in fungi, but it doesn't happen in us. So Joe likes this approach, even though it scared me to death because I had to work with radioactive stuff. But anyway, we would take tissue explants from various parts of the plants, incubate them with labeled precursors of sterols, and measure the amount of radioactivity that enters into sterols. Summarizing work that, again, you can see it's been published, is that, yes, we can use this approach to interrogate the biosynthetic rates using various precursors. And what we learned is that the spatial and temporal regulation of several enzymes in plants is not just a single enzyme as in mammals, which was kind of cool. Plants are more resilient, I guess. Who knows? And also for me, the favorite part is that jasmonate do impact sterol biosynthesis. And it makes a lot of sense because when jasmonates are produced, plants do not grow very well. There's suppression of growth. But this could be one of the ways that jasmonates suppress growth in plants. After Joe's lab, I went to Arkansas. From the introduction, Arkansas Biosciences Institute, and I worked with Helia Lawrence, who studies ascorbate metabolism. Again, I was fascinated about regulation of this network. Don't know what I was getting myself into, but it ended up being a fun experience. She's such a great mentor. And I'm showing this slide here. I'm not going to go too much into details. I want to show you this slide here because this is where I met Dr. Siddiq Abubakar, right here. So he's sitting somewhere here. And of course, we found that JA do modulate ascorbate metabolism. They touch on very many parts of this complex network that leads to production of vitamin C, ascorbate. But as I say, to me, the key was that I met Siddiq. He was a PhD student at that time. And so he interacted with me and asked me some questions since I have a PhD. And I hope he learned something, but he's here today working with us. So I came to ISU. I applied for a position here as a lecturer. And my decision really to come to ISU, I'm showing here, there are many, but there's also one person I just want to share, Dr. Walter Fair. I learned about this man during my undergraduate education at the African University. We used a textbook that he has written for plant breeding. And I met him in person in Kentucky. He gave a talk there, and I went to him, I shook his hand. I'm like, oh man, you're so famous, and you're the greatest guy on earth. I mean, all that kind of stuff. And of course, when I heard that I was going to work with him to develop some of the courses for the online program, I was even more excited. So I sent emails to my friends. I said, I'm going to work with one of the greatest breeders. And so there are many great things at Iowa State, including great plant breeding professors here, and also international engagement. All those were attractive things for me to come here. And I think I made the right decision to come here. And so I was involved in development of several of these courses. Laura Merrick was instrumental in teaching me how to write lessons. You see, I was writing papers. That's not writing lessons. And Laura knew this stuff. So I'm very appreciative of that. I'm glad that our president is thinking this, that we must prepare the leaders of our nation and the world to make the world a better place. I'm really glad. And that we must lead this. It's not going to be an easy thing, but we must lead. And we should capitalize on opportunities that the problems other parts of the world, those are opportunities for us. Be at the forefront and you take the heat, I think, or take the benefits. Helping, reaching out. So what are some of these things, the challenges today globally? There are many. And this is just, I'm showing three here that kind of are interesting to me, and I'm just thinking about them a lot. Looking at Africa in particular, Sub-Saharan region has the youngest population here, teens. This is very recent, about 2014, compared to other parts of the world where the populations are aging. Sub-Saharan Africa is facing food insecurity. Several countries here that have these young people required food assistance last year. And there's high unemployment, more than 30%. So that's to me, it's just disaster about to happen. It is happening. So NASA does a great job. I mean, they don't say the world is flat, but they put together nice images. So look at night, you're looking at the world, and you see the glittering parts of the world, and look at Africa, where there's not much light, electricity, you know. No access to electricity in that continent. I mean, I remember growing up and leaving the villages and going to the city because we were excited to see the light, you know. And I don't know, anybody here guessing what this is? Shout out. Yellow circles. Huh? Yeah? Interesting. Very interesting. Even McDonald's doesn't want to be here. I mean, don't tell them that. I didn't say that. I'm just saying that this was interesting. I would not want to just make a lot of interpretation out of this. I thought it was fun to add it to this. But these problems have consequences, global consequences. You know, people want to leave Africa and go to those parts where there's light. There's McDonald's. There's hope. And they could die doing that. You see, leaving Africa through this path here, from Libya, Algeria, and Morocco, thousands are dying. They're locked up in Libya, detention centers that the European Union is supporting right now, but they're still coming out of there, and they're running away from problems. And when they get to the place, I've arrived, I've made it, you know, I've made it. That feeling, being rescued, long trip, you know, across the ocean. But it's not as clean as that. It's not just making it. Europe is facing a big problem. And people are saying Europe is not doing enough. You're not giving enough to Africa to solve this problem of migrants. Just stop them from coming here. I mean, they're good people, and they want to help. You know? Syria is adding to this complexity of issues. So we create barriers to keep people away. What can we do? So there's a lot going on. You know, food prices, many parts of Africa, these are hotspots for food, you know, prices going up in other parts of the world, important crops. There's a lot to do. But Iowa State says that we must be at the forefront. We shouldn't be afraid. We should take this as opportunities and face this. So that's motivating. And we cannot do everything, but we can do small things. So we are there somehow. We are represented there through a Gates-funded project, education, plant breeding education, in Uganda, Ghana, and South Africa with these 3 universities. And one project here on maize improvement in Tanzania that is funded by USAID. So that I've started with the project in Tanzania. We're working with the Mikocheni Agricultural Institute and Sokoine University. And they say that we wanted to focus on 3 stresses: drought, striga, and maize, and maize lethal necrosis disease. So Thomas and I, we are co-PI on this grant. Thomas is handling the maize lethal necrosis disease because it involves a polyvirus. He's an expert on that. And I've been asked to do the drought part. And I'm not an expert on drought, but I'll try. I just want to say here I should thank Thomas quite a bit, because when we got this grant, I said to Thomas, wait a minute, we have the money, but I don't have a lab. Where am I going to do this? He goes, well, you can use my lab. Don't worry, you can use my lab. That was very helpful. And Dr. Candice Gardner said, you know, Dr. Gardner, we're supposed to send germplasm into Tanzania, GM-DH lines. She said, I'll help you. So she was there to help us. I'm very appreciative of that. And of course, this is part of the work that is ongoing, which is this work is led by Siddiq, and is part of screening germ lines for tolerance to dehydration stress, using an assay that has been developed in the Luba State Lab. Siddiq was held by a graduate student, Cici Leo. And Ushi has been quite, I don't know how to say. I mean, she has done a lot of things. Every time we go to her for management issues, lab management issues, she's there to help us. So we are getting a lot of help, and I'm sure Siddiq is also getting help from others here, postdocs and graduate students. So Sidi can say more about this because I don't know what is going to go here when it comes to association analysis. Maybe Jean-Mi can tell us because that's not me, but he's getting help. So that's I'll end there. I'll just say this is what is going on. But what I'm contributing here where I can say something is sterols. And when I was in Arkansas, I worked with an undergraduate student, Kayla, and amazingly, this student had never been in a lab, had never seen a molecular biology instrument. She was able to just put this experiment together where she would water tomato plants or not water them, and 14 days, take the leaf and look at the profile of sterols. And I say, Kyla, this is really dramatic. This change from being watered to not receiving water and having such high levels of stigmasterol, that was interesting for tomato. And in fact, when we overexpress a gene that produces stigmasterol in Arabidopsis, those plants perform better when they're not watered. So this is what we are contributing to, NSA, me, to this project. And Siddiq has gone into maize and isolated that gene. It's a cytochrome P450 called a sterol C22 desaturase. And simple, taking an Arabidopsis sequence, throw it in, get a maize, clone it, very simple. Well, I don't know, Siddiq, was that very simple? Maybe. But putting in the right cassette, transform Arabidopsis into brachypodium because we want a monograss species. And he has nice positive transformers in Arabidopsis. He's continuing the work in brachypodium with the help from Suza and Faye. And he's looking at the expression of this gene in different parts of maize. He has this, I like this. So he's continuing this work. And ultimately, we want to conduct these assays. I'm going to switch gears to the plant breeding e-learning project, unless you have a burning question. So this is a Gates Foundation-funded project involving several collaborators. We're working with 3 universities here. I've mentioned them before. Kwame Nkrumah, KNUST from Ghana, Makerere in Uganda, and UKZN in South Africa, and one large NGO that does food security work in Africa, an integrated breeding platform who have the BMS platform that is used for data, for breeding data analysis and storage. Before I proceed here, I just want to take a break, and if you're a grad student, if there's a professor next to you here, just turn around and say thank you to them, because this would not happen without the plant breeding program here. I'm really appreciative. I'm standing here taking my hat off as Iowa State, saying as an African, thank you. And you students, you should be proud of yourselves, graduate students, that you have been a part of this. Here you are. We recognize your efforts, so thanks. And of course, this wonderful agronomy club. You see, these students came to us and said, we can help you to add, to review some of these modules you're producing. Look at the technology. I mean, they went in and did some work. And it was very beneficial to us. So there's a lot of people here. I'm just standing here representing work that is done mostly by others. And so what is this plant breeding e-learning in Africa? We are part of a large project called Improved Master of Science in Cultivar Development for Africa, implemented by AGRA in Africa, in those 3 countries, and to produce applied industry-ready breeders. That's the Gates Foundation vision, who will transform and increase rate of genetic gain in Africa. And there are 9 of them at the end of the project. There'll be 9 of them. And these students would have had access to e-learning resources, so there's shortage of textbooks. They don't have high quality, current information about breeding for important crops in Africa. So they would have had access to that. We are building those resources for them. And using case studies, applied learning activities to really test their knowledge, to take them from just regurgitating information to applying it. And this content will be free. I mean, that's a huge contribution, my part, to Africa. We still have to look at the impact, I know, but. So we started by leveraging what we have through our online MS in plant breeding. And of course, we are developing additional topics. The ones in blue here, we're not. These are developed anew. I'm going to take it. So we were presented with a challenge here that was not just the material should be ready like today. Also, you should be able to use these modules, these e-learning modules, without being connected to the internet. And that's where Gretchen Anderson from the Dev Lab, Agronomy Dev Lab, came in and helped us tremendously. She suggested a platform to use. We trusted her judgment, and it worked. And it's not just that. The Agronomy Dev Lab has the experience and resources to give us an opportunity where the professor is speaking with instructional designers, and they're talking about how much text, animations, or videos should go into a module without diluting what the professor wants to convey to the students. It's just awesome to work with them. You know, you need to have that bridge to connect sometimes breeders and instructional designers, but you know, that bridge works itself out pretty well. Adapting the materials for use in Africa requires participation by the Africans. They need to be a part of this. It can't be seen as it's an Iowa State product coming to us. So we are visiting their universities. We're inviting them to Iowa State. They're working with us. We're talking to their students. We're asking questions. They're asking us questions. Slow process, but you know, I believe it's the right way to go with this. And the most exciting thing is that even though these materials have not been fully adapted for use in Africa, for their needs, I mean, they're still addressing 90–80% of the needs of the breeder from our world and other parts of the world. But they're using them. They started using the materials in the classroom. They're reading ahead of time, meeting as in groups, going to class, and spending in-class time discussing applied learning activities, where the professor can check where there are errors in thinking, gives them ownership of the learning process. So it's a new way of teaching that we're hoping is going to transform plant breeding education in Africa. And looking forward, we started distributing these materials using thumb drives, but you can't reach 54 countries. Yeah, you can, maybe FedEx to 54 countries, but so we are thinking wider distribution of these modules, and Gates Foundation is asking us to do that today, and we have the repository ready to go. Thanks again to Gretchen's team, and people can access this and see what we have in here. So I'm about to end this. So there are days, cold days, when you're walking to work and you're like, well, what's going on today? It's so cold. You know, oh, whatever is going on. Things, you know, life happens. And you ask yourself questions. For me, when I face those situations, I think of what you see right here. That's the ultimate goal. So that keeps me, that's the fuel that keeps me running. And ultimately, you know, it's that child accessing food, not going to bed hungry. It's very painful to go to bed hungry. And of course, I start with the title, the paths. I don't know what paths are, you know. There's so many paths right here. I don't know. Which road would you take? I'm just going to leave you with this. If you don't like this guy, forgive me. But I'll use my heart. Use your heart and your intuition. You cannot go wrong with that. Anyway, thanks.
Any questions for Walter? If you can repeat the question, it'll come up.
Supposed to repeat the question.
So, how would you define water use efficiency for plants?
I don't know. I think I would give you great resources though. There's a guy called what's his name from Nebraska, Lincoln. He's a soybean breeder. Yeah, Specht. He loves that kind of stuff, and I can share some of these things about water use. Yeah.
Other questions, comments?
Hey.
Talk more about the drought tolerance project in maize. What's the general strategies after that? I think you find some genes control the things, and then you're doing the podium and adoption.
Yeah.
What is the general thing you're doing there?
So our hope is that out of the GEM-DH lines, we'll find some segments that can be introgressed to lead germplasm in Tanzania. And so that involves a lot of work that you do, and Thomas and others. That's why I left it aside and not really talk about it. There's a lot of the genomics work, that needs to go into that, and also breeding. I selectively left out because Siddiqui has very interesting information to share very soon. I wanted to give him that avenue. But we are taking that approach of forward genetics and reverse genetics. I am biased towards reverse. And of course I'm working closely with Thomas in guiding us doing the forward part. Yeah. Yeah.
Would you be willing to comment a little bit on the new extension of the Gates Foundation grant that you and Mike Ritalik are working on, and sort of what the goals are for that? That might be of interest, I think, to everybody.
Thank you, Jessica. I didn't repeat the questions. Forgive me.
It's okay.
I forgot to mention about an extension of the Gates grant that happened end of last year. During our interaction with the faculty in Africa and students, we realized that the faculty themselves will need, professional development in, using the e-learning resources we are developing. So, Dr. Mike Ritalik from Agricultural Educational Studies is part of this project. Is leading us in general, creating a community of practice where the faculty in Africa can come in and work with us and others. We are not going to be insular in any way. We are welcoming other universities to join us. But ultimately, preparing the faculty as we are building the modules so that they can use them and get to their students more effectively. Yeah. Thanks, Jessica. Yes.
Can you comment a little bit about the state of agriculture in Tanzania? Like you mentioned that that's the scope of that work, the drought work, for example. I mean, what are they able to produce there? So
I'll say this, which is the simplest. Maize, 70% of people consume maize. There's rice and other crops in place, but maize is very important in that country. And there are many efforts going on, and at times is when you go to Africa today, the first thing I would say, if you want to go to Africa, first find out who is there, who is doing what. There are many organizations wanting to fund things in Africa, but you can be in a country in Africa and find that Monsanto and others with Gates Foundation are working on a drought tolerance maize for Africa right here, and here you're doing USAID project. So from development standpoint, there are efforts to address drought and other stresses, but they're either doing it in isolation. We are looking at 3 stresses at the same time: striga, the weed, and a disease that is decimating maize in East Africa right now and other parts, and I hope it doesn't get here. And of course, drought. And drought. Drought is, those who do drought, they know it's not a simple thing to fix. It's a challenge. So, did I address your question at all? Yeah. I hope. Yeah. How do I handle?
Darlene, we'll start with Darlene.
Okay, I'm just curious about how Africa accepts, the acceptance of GMOs in Africa. Widely accepted?
For consumption, it's a challenge. I just had a master's student through the online program. I supervised their creative component who worked on that question. And you have cotton in Burkina Faso, am I right? Maybe you're wrong. Burkina Faso, right? Yeah, cotton. South Africa embraces GM technologies in maize, corn. But when you come here to the sub-Saharan part here, it's still, for a long time ago, the government of Zambia said we don't want maize from the United States because we suspect it's GM corn, even though our people need food assistance. There's a lot of work, and we're very interested in that. What kind of outreach can we do? How can we combine our research here and have an outreach component where we are targeting the youth for me, I'm thinking the youth, so the secondary schools in Africa, you're having the kids, just as they come here and do DNA extraction at the biotechnology office here, you have those kind of activities, and these are the young people who are going to be leading Africa, when I don't mean that in a bad way, but when those who don't want it leave, they're the ones who will be leading this nation. So as I say, there's so much to do. I know it's a tangent, but you've hit a nerve that personally I would like to see it happening today.
Well,
Yeah.
I come from the Philippines, and it's also, well, recently the Supreme Court just didn't allow any GM research. That's why I asked the question.
So the question was about accepting GM, genetically modified foods. Yeah.
Oh, was it? Okay.
My question was if you were thinking to use biotechnology or recombinant DNA to improve, I don't know, to improve the corn or maize, for example, and what are the policies about GMO? For example, Malawi, a couple of years ago, the Minister of Agriculture of Malawi came to speak science.
Yeah.
And she was saying that Malawi is opening now to importing corn that is GMO, but I wanted to know what about the places where you are doing the research and the grants for corn.
No, how do you decide what tool to use? Well, I'm trained in that area, so I'll continue doing it. Not going to stop, but strategically, yeah, you have a valid point. That how do we enter and benefit these communities? I mean, should you use a technology that will not be accepted and you're wasting resources? But I think for teaching, if you combine research and teaching, which is, I hope that's where we are going, our part, is that what we do here, we bring scientists here and they see the process, isolating a gene, expressing it, what you go through, that by itself, I know we'll be asked 5 years, what's the impact? But some of these things are 20, 30, 50 years. Should we not start today? No, but I appreciate the question, yeah. Yeah, back, Charlotte.
Okay, do you have data mechanisms to assess the impact of this e-learning?
Thank you. So mechanism to assess the impact, and microitalic is helping us on that. So this extension part of the project includes that, creating a mechanism for assessing the impact, actually baseline information so that we can come and look at whether you've made that impact or not. That's a good question, thanks. Yeah.
Other questions? Yes, Nicole.
You have to hear that it's not necessarily quantity that's a problem, but transportation. You can't get food to where it needs to be. Is there a lot of work going on with things like food products, food vegetables, grains you can handle being shipped in substandard transportation methods, can handle long periods of time without refrigeration, things like that. So you don't have to necessarily keep driving up production. You can use what you already have more efficiently.
Yes, you're right. It's not just production. So I want to be correct when I say this. Production is important. I mean, you're looking at 1.5 tons per hectare. You want to push it up. I mean, it's dismal when you look at maize yield in Africa right now. But it's an opportunity because you haven't even hit the exponential part. But if we think ahead of time about this problem, but they've been there. So 30 years ago, we heard of those same problems you're mentioning. And this is why, to be honest, it's critical to have this kind of conversation at the table when we're talking about funding production, but have someone like that coming in with that. Just think a little bit, what about this part? And I don't know how you can get donors to want to do an integrated kind of an approach. But yeah. Thanks.
Can you comment on the maybe second phase or next phase of the GATES program,
Yeah.
how do we train those teachers to take advantage of this e-learning modules? As I started to teach the class, I found out that I have to teach myself first
Yeah.
before I can have a good grasp of how to
Yeah.
facilitate the student learning. They're textbook ready,
That's right.
and there's now, ignoring the price issues.
Yeah.
Now we change everything to e-learning modules, which is more accessible to students, right? And then there's also still a need for professors, faculties to guide the learning process.
Yeah.
And then if we have this e-learning modules being used by those African, professors, instructors, how do we train them to understand their challenge or their role,
Yeah.
adapt the role, improve the role, change the role? That seems so critical because unless we train the teachers to be a better teacher, to use this material, otherwise we're just a substitute of a textbook or similar or slightly improved. We might not reach the potential. That's my concern. Yeah.
No, Jianming, you're right. You're right. And this is why this small extension that Mike Ritalik from Ag Education is going to lead us in. You see, Mike believes that, and he says this openly, he says, you can have the best e-learning modules, but if teachers don't want to use it or if they're not prepared, you're not going to make that impact. So a little bit of the approach that he's going to use is to bring the faculty here at Iowa State.
The second phase. Okay.
To bring them here. Actually, it started already. This is the first phase that has just been extended. But GATES works in an interesting way. But they would come here for symposia. They would have an opportunity to attend classes here and see our faculty are using the e-learning for the online MS, delivering the materials. And he's going to work with them to identify what are the professional development needs for them. I think when we came in and you were part of this project, Jianming, is that there wasn't a lot of thought behind the teacher part or assessment part from GATES, but we've traveled together and now they are listening and they want to do it. Because you can't just have e-learning without the teachers. So, but you're right. Yeah, thanks.
I think one programming note sort of to bring along with this is March 1st, actually, we're adding a seminar to the schedule that actually will have many of the educators in Africa come and informally/formally discuss their use of these materials. So if you're at all more interested in this, do plan to come on the March 1st. And I think we're going to have basically small presentations by some of the faculty from the different universities, and then some informal just discussion. I think for all of you sitting out in the audience, if